Hello students, how are you? How is studies going on? I hope all of you are doing well.
In today's class, we will read about sexual reproduction in flowering plants. To know the sexual reproduction in flowering plants, at first we have to know about a flower, a typical flower. So this is a diagram of a typical flower where you can see that this is the stalk of the flower, which is called pedicel, followed by your thalamus, which is the receptacle, and this is the calyx, the first hole of the flower. Then is the corolla. Next is your androecium, and this is the gynoecium. So out of all the four holes of the flower, these two are the essential holes of the flower which takes part in the sexual reproduction. The rest two holes, that is your corolla and the calyx, these are the non-essential holes of the flower. Their main purpose is to attract the pollinators for the purpose of pollination. As we have read in our previous class, that sexual reproduction comprises of three different stages: pre-fertilization, fertilization and post-fertilization. And in the pre-fertilization, the first stage is your gamete formation, that is gametogenesis. So gametogenesis here will comprise of formation of pollen grains inside this anther, which is called as your Microspore Genesis. And the next part will be the formation of the megaspore, which will be inside this ovary, which is called as your Megaspore Genesis. So let us start with Microspore Genesis. This is a cross section of the anther. The anther is a bilobed structure. If you further see it in the cross section, you will find that each lobe is having two thecas. So moreover, you can see that there are four chambers. In this chamber, if you closely look into, then you will find that there are certain cells. This has been highlighted over here, which are called as your microspore mother cell, which are the potential cell which will undergo further meiosis to form the microspores or the pollen grains. But before that, we have to know all the other layers of this sporogenous tissue which is present in the anther of a male flower, a male part of the flower. So the outermost layer of this, that is your the outermost layer is called as your epidermis, followed by the endothecium. Next, after the endothecium, these dotted structures with the nuclei, which is present over here, this red color nuclei. This is the tapetum layer, which provides nutrition to the developing microspore mother cells, which will undergo meiosis to form the tetrads.
Inside the microsporangium, each cell of the sporogenous tissue is capable of giving rise to microspore tetrad. Each one is a potential microspore or a microspore mother cell, which is called as your PMC. Now, this process of formation of microspores from pollen mother cell by the help of meiosis is known as your microsporogenesis. The microspores, as they are formed, are arranged in a cluster of four cells. When they become mature, they are dissociated from each other and they develop into pollen grains and are dispersed into the surroundings. Now, a typical pollen grain is having an outermost XII and the inner intine. The XII is discontinuous and is variously designed in case of various kinds of pollen grains. They are having a chemical substance inside them, which is called as your sporopollenin, which is highly resistant to any kinds of chemicals or any degradation due to enzymatic activity, and they can be even fossilized also. Now, the pollen, the intine is intact, of course, and there are some germ pores on the XII through which the intine can develop the pollen tube when they reach the stigma of the desirable species. Now, the pollen, inside the pollen, since it is haploid, it at first undergoes mitotic division, which results into formation of two cells which are very unequal. The bigger one is called the vegetative cell and the smaller one is called as your generative cell. In case of most of the angiospermic plants, the pollens are shed in this two-celled condition. But in some cases, it has been also noticed that the generative cell is again undergoing the second mitotic division before it is released So that is called as a 3-cell condition. The pollen grains sometimes can cause allergic reaction. The viability period of the pollen grains also differ from plants to plants. In some cases, like wheat and rice, they are just viable for that of the 30 minutes, whereas in case of plants like pansy or the plants which belong to that of your Rosaceae family, they are viable for months. So this pollen grains since they are important for the process of pollination, resulting into formation of the seeds for the next generation to be stored. They can be even stored in that is your liquid nitrogen, and at minus 196 degrees Celsius, to be used further for the plant breeding programs. And this phenomenon of preserving the pollen grains in liquid nitrogen is known as your cryopreservation.
The next part of sexual reproduction in flowering plants includes another process of gametogenesis, that is formation of megaspore in the ovary, which is called as your Megaspore Genesis. Now this is your typical ovary where the process of Megaspore Genesis takes place. Now in the ovary, if we go deeper into in the ovary, there is a structure called Megasporangia or Megasporangium, which is nothing but the ovule which is attached to the ovarian wall by means of placenta. You have read it in class 11 about placentation. I'm not going for that. Now the ovule is connected to the wall of the ovary by means of placenta through a stalk-like structure. Now this stalk-like structure is known as your funicle. And this is the funicle through which the Megasporangia is connected to the ovarian wall. And this is a typical anatropous ovule. Inside the ovule, the ovule is externally protected by two integuments, and then is a nutritive layer which is called as your nucellus that gives nutrition to the developing embryo or the other cells which will be forming later on in the embryo sac. Now the integuments which are encircling the embryo sac, they are not completely encircling. There's a pore, and this pore is called as your micropylar end, and the portion which is just opposite to that of the micropylar end, that is called as your chalazal pore. So depending upon the arrangement of the micropylar end and the chalazal pore, the ovules are named differently, but in our book it is anatropous ovule, so we will be reading about that. Now there are various stages which takes place inside the that is your ovule, the changes which happens in the embryo sac. A typical angiospermic flower, inside it, the formation of the megaspore takes place from the nucellus cells. From the micropylar end, one of the nucellus cells forms the that is your Megasporangium mother cell, which undergoes meiotic division. Now when it forms meiotic division, it forms 4 haploid cells, and out of the 4 haploid cells, only one becomes the potential or the functional megaspore cell. The rest 3 degenerates. Now we will talk about a mature embryo sac, what events takes place inside the embryo sac. So one of the megaspore, which is the functional megaspore, inside it, since already the nuclei is haploid, it has undergone meiosis, so it will at first form mitotic division. Mitotic division resulting into formation of 2 cells that will undergo further two more mitotic divisions. That means in total there will be 3 mitotic divisions. So at first from 1 to 2, then from 2 to 4, and then from 4 to 8. Now these 8 nuclei which are formed over there, they will be arranging themselves in a particular fashion. 3 will be going to the chalazal end, 3 will be going to the micropylar end. Those 3 which will be going to the chalazal end and the micropylar end. Now at first the division is free nuclei, they will not form any wall, but later on wall formation will take place in them. Now this will be called as your antipodal cells, and these 3 are called as your egg apparatus. In this egg apparatus, these 2 are the synergids, and this one is the functional egg. Now the synergids function
as you can see, these two structures, these are called the filiform apparatus, which will be guiding the pollen tube towards the micropylar end for the process of fertilization. The central two nuclei, this will be called as your polar nuclei. This nuclei takes part in the formation of your endosperm, whereas the egg nuclei takes part in the formation of your embryo. So a typical angiospermic embryo sac, although is 8 nucleate, you can see 1, 2, 3, 4, 5, 6, 7, 8, but it is 7 celled because 1, 2, 3, these 3 cells, 1, 2, 3, 6, and this one 7.